This is the mini lecture for cell anatomy. So before we even begin to consider what is inside a cell, it's important to think about the relative size of cells. So this diagram helps us do that. To the left on this diagram, there are things that you can see with your unaided eye, and these are macroscopic items. In this course, whether in person or online, we're going to use a microscope to view pretty much everything. There are many websites, and two of them are listed at the top right of this slide. And these websites, they kind of walk you through a relative scale. They're kind of fun. You should take a look if you can. You get a sense of the size of cells, their components, and maybe some things you might be familiar with, like yeast or DNA or carbon atoms. But these websites show you all microscopic things. Another aspect of the external anatomy of cells is their surface area to volume ratio. And this is the comparison of how much covering there is to how much insides. So your cutaneous membrane, or your skin, is equivalent to your surface area, and it is quite large. Your insides, your squishy organs, your muscles, your bones, those are your volume. So you want this ratio to be large most of the time so that exchange with the external environment is efficient. I like to think of my cats. On cold days, they curl up, and one of them even like burrows into blankets. And their volume is fixed, right? So as she curls up, she reduces her surface area and she reduces her heat loss to the environment. But then on hot days, or when we have the wood stove going, they get like as close as they can to the stove, but they stretch out, right? And they try to increase their surface area and increase their heat loss or prevent what's really a cooked kitty. There are so many different kinds of cells. And one way we create large categories of cells is to compare simple cells, called prokaryotes, to complex cells, which are called eukaryotes. Prokaryotes are things like bacteria and other fascinating organisms that are called archaea. Archaea are simple-celled organisms, and they live in extreme environments like volcanoes and hot springs and really salty lakes. We're going to consider the more complex cells called eukaryotes or eukaryotic cells. These cells have what are called organelles. And yes, organelles are to cells what organs are to your body. So we explore plant cells and we explore animal cells like those pictured here. But I want to remind you that there are other eukaryotic cells of fungi and these baffling organisms called protists that are listed on the left and right here. We discuss these organisms in the next part of this course, so that would kind of be like next semester. All right, so let's zoom in and let's take a look at the components of a eukaryotic animal cell. And this is like zooming in on your body and looking at your liver that detoxifies your blood, your brain that controls your voluntary and involuntary muscles, and among other things, your digestive system that breaks down molecules. Whenever we start looking at these little components called organelles, every book, it always starts at the nucleus first. And that's the reason behind this. Eukaryotic cells have a nucleus. Prokaryotic cells do not. So if you're doing a microscope exercise and you know you have a eukaryotic cell if you have a nucleus. I like this picture here because it shows you a drawing representation of the nucleus, but there's this cutout of a microscopic picture of it too. So the nucleus, its purpose, it houses DNA, right? DNA comes in many forms, chromatin, chromosomes, and other smaller divisions, but it's all found in the nucleus. The nucleus is double wrapped with what is called the nuclear envelope. It's like a plasma membrane with only these openings or pores. And no other stuff can get through, just DNA or just RNA, no other stuff like proteins or cholesterol. The DNA holds the instructions for life, or the recipes of proteins, and proteins carry out all of your functions of life. So it's like the recipe book, and you are the collection of recipes that it can make. All right, while the recipes or the instructions for the proteins are locked up in the nucleus, the machinery for making the proteins is in the cytoplasm, or the gooey substance of the cell that's inside the cell membrane, but outside the nuclear envelope, so like in between the two. You have to think of the nucleus like the reserve materials in a library. Anybody go to the library anymore? Reserve materials can be used in the library, but they can't be removed from the library. So things like encyclopedias and dictionaries. You can copy the information in the reserve materials, and you can copy the information in the nucleus. And you can get the information out of the library of the nucleus. But you can't remove the DNA, you can't remove the books. The copy of the DNA, it's called mRNA. And you can kind of see here on the bottom right corner as this like pink ribbon. It's being read by this brown double-parted thing called a ribosome. And the ribosome is making a polypeptide, which will be folded into a protein. And it's making that according to the instructions on the copy of the DNA. We go into this process in much more detail in later modules. Ribosomes can be free-floating in the cytoplasm, or they can be bound to this internal network called the endoplasmic reticulum. So free ribosomes, they usually make proteins that'll be inserted into the cell's membrane, whereas ribosomes bound to the ER, they usually make proteins that'll be shipped away from the cell and used elsewhere. What is the endoplasmic reticulum? Other than like what it looks like, a maze of tubules, right? What is it? So let's get some root word exercises here and dissect this name. Endo, you know what that means. It means inside, right? And plasmic, well, we must be referring to something that's inside the plasma membrane. So we have endoplasmic, inside the membrane. And then reticulum is a word that means network or netting. And that's what this organelle looks like. We consider the rough and the smooth endoplasmic reticulum, or ER, as separate organelles, even though they look alike and even though they're attached. The smooth ER is a lipid-making organelle. Its main role is in detoxification. It makes lipids and they surround toxins, allowing for their removal. Your liver cells, which are called hepatocytes, they filter toxins from your blood, and they tend to have lots of smooth ER in them. Actually, alcoholics with a tolerance to alcohol, they have tons of smooth ER in their hepatocytes, trying to eliminate all of the alcohol. Cirrhosis of the liver, which is just an inflammation of the liver, it can result from this overabundance of smooth ER in all of the cells. It's basically they're crowding out everything else. The rough ER has tons of jobs, but most of the jobs have to do with the building of a protein. The rough ER, it plays a role in folding the polypeptide chain, and the rough ER, it can also make these little bubbles of phospholipids that are called transport vesicles. And the transport vesicles contain the protein so that it can be transported to the next organelle in the process of making and shipping that protein. But the main difference between these organelles is that the smooth ER, it gets all of the girls. All right, so the Golgi apparatus has been called many things in many different books. The Golgi body, the Golgi complex, it's all the same thing. If it makes you feel better, we can just call it the Golgi. I don't care. It's all the same thing. I like this organelle because it looks like a stack of pita bread if you cut it and look at it from the side. Think of this organelle like a conveyor belt. A protein that's contained in one of those vesicles from the rough ER, it enters on like the receiving side of the Golgi. And then As it passes through the Golgi, it kind of gets labeled for its final destination, and then it gets popped out the other side, kind of like the shipping side, in a new transport vesicle. The Golgi makes vesicles for other reasons too. Some vesicles don't contain proteins, and they leave the Golgi, and they get inserted into the lipid bilayer for repair. And then some of them actually turn into our next organelle, which is called a lysosome. Lysosomes they're basically little stomachs that float around the inside of a cell and digest things like broken down organelles. The term lyso means to split. Rumors have it that it's the origin of the brand name Lysol. It splits bacteria and germs. Lysosomes originate from the Golgi apparatus, and they contain digestive enzymes. So an interesting but very upsetting disease called Tay-Sachs comes from a malfunction of these lysosomes. In Tay-Sachs disease, the lysosomes do not contain an enzyme for lipid breakdown. This is a genetic disease in which the DNA code for the enzyme is not present in the individual. The lysosomes become engorged with the lipids from the inability to break them down. Basically, they become a storage factory for them. And as the lysosomes fill and fill, they crowd out the other organelles of the cell, and essentially it becomes fatal for the cells and then the organisms. Getting back to lysosomes in general, it's important to note that lysosomes are only present in animal cells. Vacuoles, on the other hand, are present only in plant cells and protists, but we're going to kind of leave them out for a little bit. This is a bubble. The vacuole is a bubble for storage of minerals in water. It's like a water balloon. On the left here is a protist with a vacuole that contracts and expands, and it creates pressure differences for movement. In the plant cell on the right, the vacuole is filled with water when the plant is well watered, and then the vacuole empties when the plant is wilted. Another organelle that's present only in plants and protists is the chloroplast. Mention of this organelle usually makes students think of photosynthesis. And yes, this is where photosynthesis occurs in two steps. The first step uses light, and then the second step uses carbon dioxide. Chloroplasts have this inner and outer membrane surrounding what looks like stacks of coins. The spaces between these structures are used to create gradients that harness energy. The green color that we associate with chloroplasts is called chlorophyll, but we're going to learn a little bit more about pigments in later modules. Chloroplasts are one of the two energy-converting organelles in a cell. Chloroplasts are, unlike the Golgi and ER, they don't contribute to the making of proteins. They contribute to the conversion of energy. The other energy-converting organelle is mitochondria. You have to say it like that. It's a common mistake to think that chloroplasts are present in plants and mitochondria are present in animals. But mitochondria are present in all eukaryotic cells. So we're talking about plants, animals, fungus, and protists. Once the plants make glucose from photosynthesis, it's then used by the mitochondria to generate ATP. For animals, we don't get the glucose from our chloroplasts. We don't have them. We eat the plants to provide the glucose to make ATP. Either way, it's a mitochondrion that makes ATP. Much like chloroplasts, there's this setup of membranes, and it allows the creation of gradients. The energy in these gradients can be harnessed to make ATP. Kind of like the Hoover Dam, it converts the energy of running water into electricity. While creating ATP, mitochondria consume oxygen and glucose, and they produce carbon dioxide and water as waste. If you're a plant, that's awesome. because you can then use these things to harness the energy of the sun and to create glucose, and it's this huge cycle. All right, so this is like a completely random slide. It's like the kitchen drawer of organelles. Like, here's all the other stuff. All right, so there are these little barrel-like structures up here on the right, top hand right, and they're called centrioles. These are used to make other organelles that are needed, like centrosomes that direct mitosis, cilia and flagella, which are rotational organelles. So a flagellum, which is present only on human sperm in the human body, it's not a tail switching side to side. It doesn't do that. It rotates with a smooth and constant motion around and around. Cilia, which are present on your trachea, your windpipe, they beat in unison to trap particles and then move them upward. Down here on the bottom right-hand picture, cilia look like feathery appendages on cell surfaces. You can see an enhanced view of a cell membrane here. On the top is the outside of the cell, and on the bottom is the inside. Inside a cell is what's called the cytoskeleton, which seems kind of boring at first. Yes, yeah. It is like our skeleton, providing support. But the cytoskeleton in individual cells is an intricate network of filaments along which proteins are transported. It's like a highway. Check out the video link at the top right of the slide once you're done with the PowerPoint. The link is a TED Talk with some projected views of what the cytoskeleton may look like. The outside of a cell, especially in your body, is also an intricate network of proteins like collagen with cells suspended throughout. Your lymph nodes are a network of fibers that your white blood cells move through while they scan other cells that are also moving through that matrix. All right, take a look back on the groups of organelles that we talked about. We had organelles like the nucleus, the ER, and the Golgi that took part in protein manufacturing. Can you locate those on this diagram? Say yes. Yes. We also had mitochondria and chloroplasts, which converted energy. No, there's no chloroplasts on this diagram. And then we had some appendages and like an inner highway of support. So can you tell me what each one does? Can you label them? Get organized for the quiz. You know there's going to be like a matching, and it might be the name of the organelle and the picture. It might be the name of the organelle and its function. It might be the picture of the organelles and then its function. Just get ready. Know the anatomy and the physiology.